Dissolution testing is a performance testing for the pharmaceuticals. Dissolution testing provides the valuable information regarding the in vivo behavior of the formulation. Dissolution test is a in vitro test to understand the in vivo behavior of the formulation. And dissolution profiles can give you information and correlation with the pharmacokinetics of the drug substance in the formulation. Or in simple way, you can say that the pharmacokinetics are related to the dissolution profiles. There is a relation of the test in vitro and the in vivo. So there is a correlation between dissolution profile and pharmacokinetics. You might be knowing the term IV-IVC. So that is in vitro in vivo correlation. And this is the topic for the present video. I have named it as or term as or topic name as dissolution profiles and pharmacokinetics. But the meaning is same and the understanding will be same. I welcome all of you to this video, and after watching this video you will understand the importance of dissolution profile for the pharmacokinetics or ADME behavior, or you can understand how the dissolution profiles are related to the pharmacokinetics of the formulation. Let's start with the information for pharmacokinetics and link between the dissolution profiles and the pharmacokinetics. So the dissolution profiles give you information regarding how the formulation behaves in vitro or how the formulation release the drug in vitro. The success of the drug depends on both its formulation and how efficiently it reaches the bloodstream. Dissolution testing directly impacts the pharmacokinetics. Now coming to the dissolution profile, you might be knowing dissolution Q point. So dissolution Q point is the measurement of percent drug release at single time point, while the dissolution profile means how the drug is getting released or how the drug is getting dissolved at different time points. Dissolution testing simulates how the formulation releases or how the API get dissolved from the formulation in the body. Dissolution is the term that you can understand by in vitro also and by in vivo also. Dissolution in vitro profiles helps you to understand how the formulation will behave in vivo. It measures rate and extent of the drug dissolution. For example, 30% in 5 minutes, 90%, 95% in 30 minutes. Generally, the quality control test for dissolution involves single time point, while to have the understanding about the pharmacokinetics behavior, it is done by the dissolution profiling. So dissolution profile ensures batch to batch consistency and it predicts the in vivo drug behavior. Now coming to the information on how dissolution affects pharmacokinetics. So absorption is affected by the dissolution rate. Slow dissolution may lead to incomplete absorption. Peak concentration, that is Cmax, is also related to the dissolution. Faster dissolution increases peak drug levels, influencing efficacy and safety. Time to peak, that is Tmax. Slower dissolution delays therapeutic action, while the faster dissolution will have the faster Tmax achievement. Bioavailability, that is AUC. So poor dissolution lowers the drug exposure, affecting the treatment outcomes. There should be specific rate of the dissolution, and the dissolution should be complete. So rate of dissolution or dissolution rate and dissolution extent is directly related to the Cmax and AUC. Example, a tablet with 95% dissolution in 30 minutes ensures rapid absorption, while a tablet with 50% dissolution in 30 minutes may require higher or more frequent doses, or it may have delayed Cmax. Now we will see the role of discriminatory dissolution profiles. So discriminatory dissolution means the dissolution which can detect meaningful differences in the drug release due to formulation changes, manufacturing changes, or storage changes. I have made many videos on discriminatory dissolution. You can go through those videos to have a complete idea about discriminatory dissolution. Now see the impact of this on drug PK or pharmacokinetics. Formulation changes, excipient variations can alter Tmax and Cmax, reducing the efficacy. Some formulation excipients are there which can improve the solubility or which can decrease the precipitation of the API in the GI, and through this, these formulation excipients impact the in vivo behavior. Generic bioequivalence. Generics must match reference drug dissolution profiles for similar PK performance. Controlled release products. Deviations can cause dose dumping or suboptimal drug release, affecting the safety of the formulation. So both here, both things we should understand regarding the immediate release and modified release formulations. Real world implications and why it matters, because it has impact onto the industry, it has impact onto the public health. Impact on industry. Generic drugs. Minor dissolution differences affect therapeutic equivalence. Formulation strategy. Enhancing dissolution improves bioavailability of the poorly soluble drugs. Many technologies can be tried to improve the dissolution, to improve the solubility of the drug, and by that we can also enhance bioavailability. Therapeutic outcomes. Rapid dissolution profiles are required for acute treatments like analgesics or rescue medicines. Controlled dissolution maintains a steady state drug levels for the chronic conditions. For example, asthmatic conditions can be treated with some controlled release formulations. Why it matters? R&D teams, early optimization prevents late stage failures. So R&D team should be having a good understanding about the in vitro in vivo correlation and how the dissolution profile affect the PK parameters. Quality teams to ensure product consistency and predictable patient outcomes. Clinicians should understand PK variability in the drug responses. And in conclusion, dissolution profiles plays the critical role in the pharmacokinetics and overall drug product behavior in vivo, and thereby it affects the safety and efficacy of the drug products. Thank you for watching the video.